Okay, welcome everybody to our final session of the day, where we get to continue our monitoring, policy, and innovation session. Jenna will be our moderator for this session again. So Jenna, I'll pass it over to you.
All right, Maddie, and back by popular demand, I have some more Valentine's Day cards for everybody to share quickly. And so if you can see my beautiful PowerPoint here, I never want to purple loosestrife you, never want to lose you. So that's, we don't want to lose any participants by the end of this. I know it's the last session of the day. We've got I'd grow anywhere for you. I'd go anywhere. So Phragmites, a nice clear picture of that here. Again, you have my heart in giant knotweed, so a play off of the last knotweed, and of course you have invaded my heart. So all of you here today are part of that, so we're lucky to have you here, and we will get going with our session. I'm pleased to introduce Isabelle Aubin as our first speaker for this session.
Good, thanks, Jenna. So I'll just share my screen. Good, so you see my screen, all good?
Yeah, everything looks great.
Okay, good. So yeah, thanks everyone, thanks Jenna, and thanks everyone to be there at the end of the day. So my name is Isabelle Aubin. I'm a research scientist in forest ecology at the Greater Forestry Centre. And I'm here today to present our work on the vulnerability of Canadian forests to exotic pests. But first, before starting this presentation, I would like to present the team working on this project. We are vegetation and pest ecologists and modelers working together with the support of biologists and data specialists, so a great team to work with. So the introduction of exotic pests is not new. Our forest has in the past faced massive tree mortality after the introduction of invasive pests, which can just take up which disease and maybe more recently. What is new now is the rate at which these introductions occurs. With the increase of international exchanges, the introduction of exotic pests is becoming more frequent, and our forests are also projected to increase drought events, an important change in their growing condition with climate change. So this is these important changes with climate change make trees more sensitive to pests while we are having an increase in number of pests and insects. So this is really making our forests more vulnerable than ever. So we had the idea of this project 2 years ago at the Forest Test Program. After hearing about all the new pest species coming in, we felt that we needed an integrated portrait of the risk that pose all these new introductions. We thought that would be helpful to understand their impact on Canadian forests. We have the question of how much of Canadian forests are at risk of being affected by these exotic pests. Are there vulnerability hotspots, region less affected, species less affected? So to get a bit of a sense of those questions and obtain an integrated assessment, we combine the distribution of 40 exotic pests of concerns that are currently extending their distribution in Canada, with the distribution of the 37 most abundant tree species in Canadian forests. So to do this work, we use the following layers of information: the stand composition, which are the tree species distribution and the estimate of their biomass that we obtained from the work of the colleague André Baudouin from the Laurentian Forestry Centre. Then we have the pest distribution. We have both the current distribution that John Pedlar wanted to co-author, retrieve the occurrence data from each of the selected pest species from GBU provincial data set interpretations. It's a big work. And we were also interested to assess the potential risk in the near future. So we have done some, again, John have done some projection of the pest species climatic niche onto the Canadian tree species distribution using the maximum species distribution model. And we've done that for a projection that we have in the near future, so 2021 to 2030. And the last layer of information we use is the severity index that we will just present in the next slide. Okay, so to be included, the pests, I told you we selected 14 exotic pests. So to be included, this is the criteria we had. First, it needs to be exotic. broadly defined here as a species introduced into an area outside its normal distribution. We are including here a species extending its distribution to new areas in response to climate change and treating here a long-term climate risk. And also to be included in our analysis, the pest should be suspected of inducing significant tree mortality on at least one of the 37 tree species we are working with. And the pest also, we only selected pests that were at the introduction phase of the infestation. So the pest should be relatively recently introduced or with very high potential of being introduced in the next years. We included pests in Canada that were in Canada for decades, only when they're still moving actively through the Canadian landscape. And we excluded all introduction that are now widespread and under natural biological control, such as the large Saiwa. We include fungus only when they were introduced by when they were at the dispersal vector by an introduced insect. So like the beach disease, the beach bug disease, and so we're not included here the whole field. So with these criteria, we had 14 pest species that were included. And we've included in our analysis also characterization of the severity of the infestation for each of the combination of species, of pest species and tree species. So to do so, we carried out a literature review and we consulted experts to characterize the potential insect damage on each of the 37 tree species. We used a classification that was developed by Meta in 2019 that we adapted to the Canadian context. So our index goes from low severity, where only mortality of weakened trees is likely to occur, to a high severity of a severity class of 6, where functional extension of the tree species is likely to occur. So the 14 species we included represent a range of severity. It includes, for instance, the brown spruce longhorned beetle, which is currently found in Nova Scotia, where it causes mortality mainly on weakened spruce trees. You have a map of this current distribution, and here you have a map of this climatic niche, which with the transport of infected wood, this pest could potentially spread and affect species in other regions of the country, as we can see in this map. We also included beach bark disease, which is an insect fungus complex, so a fungus which is carried out by a beach tail insect. The beach bark disease has a higher severity than the brown spruce longhorned beetle, so it is currently causing extensive mortality within the beach population in eastern Canada. And this pest is present in Canada for several decades, but is currently moving quite fast through the beach tree ring in Quebec. We also included species with higher severity, very high severity, like the emerald ash borer, which is currently causing massive mortality of ash species. In the last 20 years, this insect had a quite rapid spread, mainly via the transport of infected firewood. This is an insect that we are working in my lab. We're following ash stems to see a bit of the fate of the ash stems and the outbreaks of emerald ash borer. So this is an insect I'm a bit more familiar with. So this insect has a high severity. It has the potential of bringing functional extension of the ash species. So, but usually we say tree mortality rate is 98%. That's what is reported. But in reality, personally, I never found any mature trees that survive. We only found some trees that are likely to die. We're still searching for the resistant one. But the thing is that ash is having important regeneration, and we're currently following the fate of this regeneration. So that's an interesting story to follow. So no time to present you all the 14 pest species, but this was just giving you an idea of the type of species included in these maps. So this is the map we have developed. It's a map combining the current distribution of the 14 selected exotic pest species with the biomass of the 37 most abundant tree species in Canada. So of these 37 tree species that we examined, 24 of them can be killed by at least one of these 14 pest species. So this map here shows the areas where current tree species biomass are exposed to exotic pests and the weighted average severity of their insect damage. So we use here a legend with color code. I think I can show this. You see it's a two axes. In the x-axis is the tree biomass of trees that are at risk of mortality by at least one pest. And in the y-axis, it's the severity index that range from a low severity index where it's green to green, and a high severity index which is orange to purple. So in this map, we can see two hotspots of vulnerability. The pink of the map in the west coast. This is where you have a high tree biomass that is at risk of a high severity. So it's mainly caused by the mountain pine beetle, but also there is also several other pests that are included included into this map, although less extensively distributed. Some adelgids, sorry, English, and coleophora species as well. So it is important to note also that we didn't include the historical distribution of the mountain pine beetle in this map. We only included where the mountain pine beetle was considered exotic, so outside of this historical range, outside is not normal distribution. And yeah, this is the first hotspot, and the second hotspot, although representing less tree biomass at risk, it's in the East Coast. You can consider it as well as a hotspot, especially along the seaway and the border with the US. So the identification of these hotspots may be useful to determine regions that may require closer supervision or to get a view of what's happening, and also try to limit the potential impact and get help a bit for the decision of where to prioritize action. So I'm adding here a second map at the right with the projection of the potential progression of exotic pest infestation in the short term, so 2021 to 2040. So this map is based on the climatic niche of the 14 pests that the current and so we have combined the 14 pests and their climatic niche and the current distribution and biomass of the host trees. So it is important to note that the projection that we're using here are not constrained by the pest dispersal rate. So it does not inform on the capacity of insect to reach those areas. It just tells us where the host and the climate is adequate for the pest for its development. So we can still see that the tree biomass potentially at risk remains principally in the two distinct areas, the east and the west coast, but with an important potential of expansion. In Ontario, the tree species at risk include the ash, the hemlock, which are at risk of functional extinction, and the beech, also which face a high risk of mortality. But also, it can also include the pines in Eastern Canada, because they may be also at risk in the future if there is a further expansion of the range of mountain pine beetle to climate change. So of these 37 tree species, we said 24 were at risk. But in the east, what is left without this? So we have the maple, the poplar, the basswood, and the cedar. But for maple and poplar, we have been very close to a different story. As we know, the Asian longhorn beetle, a wood borer pest which attacks maple, poplar, birch, willows, several species, have been found several times in different ports in North America and since the 90s. And quarantines in port are currently in place to stop its spread. And this insect has been found and successfully eradicated from the city of Mississauga, Toronto, into a tree and again into a tree. So little is said, I thought this was important to mention that little is said about this successful eradication. They don't make the headlines that much because we avoided the worst. No story to tell, the disaster scenario was avoided. So it doesn't make the front page of journal, you know. But So the more effective we are at avoiding the worst, the more difficult it is to demonstrate the importance of this work. So that is why we wanted to map the risk that ALB would have posed if we had missed it in 2013. So the map on the left is the one I showed earlier, combining the climatic niche of the 14 pest species with the distribution of biomass of their tree hosts. In the right, I'm adding here a map with the climatic niche of the Asian that we have the 14 species, plus the Asian longhorn beetle and the biomass of its hosts, so the maple, the birch, and the poplar. So this map showed that the expansion of Asian longhorn beetle in Canada's forested landscape would have put an additional 6 tree species at risk, representing a potential for its biomass at risk estimated at 4.9 million tons. So these maps provide evidence of the importance of solid detection in the registration program that we have talked a lot this afternoon in different parts. So this program, we can only say that they are very important because the decades we gained from these registration program, even if one day the insect get through the landscape, it will give us a time for developing methods to control the pests and better manage their impact. So those decades are important. A good example, even if it went quite fast, for emerald and borer, the information we get from the southern Ontario is useful for it's knowledge that is useful for better prevention of the spread of VAB and also better mitigation of the impact in the Northland sector. So yeah, I'm at like 15 minutes, so time is running. I will stop here, but Ananda and myself are available for questions. Thank you.
Thanks so much, Isabelle. Great presentation. I think it's a little bit alarming to see everything, but you know, when we say invasive species are the second greatest threat to biodiversity, this is, you know, the context to that. So really helpful to have your presentation today. We do have a minute or two for questions. If anybody has anything to ask, please feel free to just pop it in the Q&A box. Yeah, some great information here. So definitely a great opportunity to ask some questions if anybody has any. Yeah, we'll just give it a minute.
Day like that, eh? It's
Yeah,
Valentine Day.
Yeah, for sure. Yeah, we're getting close to the end of the day too. So yeah, yeah, for sure. Maybe I'll ask if you don't mind watching the chat and everything, just in case anything comes in. But we can always move on, and if you don't mind, yeah, just keeping an eye out. That'd be great.
Yeah.
All right. Thank you so much again, Isabelle.
Thank you.
All right. So we'll move on to our next speakers. So we've got 2 presenters coming up here. So we have Peyman Saidi, and we also have Matt Bolding from Ducks Unlimited. So they're going to be talking about European water chestnut. So if you're both ready to get going, I think we're good to go, right on schedule here.
I'm going to share my screen. Can you confirm that you can see my screen?
Yep, looks great. Thank you.
Good afternoon, everyone. Today, I'm pleased to be here alongside Matthew Bolding from Ducks Unlimited Canada. And we are here to present our recent collaboration between Ducks Unlimited Canada and Saiwa on the automatic detection of European water chestnut from drone imaging. Matthew will discuss the project's significance first, and then I will follow with presenting the technical aspect of the work. Matthew, over to you.
Thanks, Peyman. So I'm just going to give a quick introduction of European water chestnut for those of you who aren't familiar with it. This is a floating aquatic invasive that forms these really distinct rosettes, like you can see in the sort of inset images here. European water chestnut ticks all those standard boxes for an aggressive invasive plant. The larger image, you can see a big infestation in the Erie Canal. It spreads really fast. It blocks light penetration under the water, chokes out native plants, is really bad for waterfowl, for fish, and other wildlife. And it also produces these gnarly chestnuts, these little seeds that actually like are kind of a hazard if in shallow water and they're there. And this is definitely a species of concern in Ontario. Water chestnut was originally, like a lot of invasives, introduced to North America as an ornamental plant way back in the 1800s in the US, and sort of creeped its way north slowly until I think it was observed in Ontario in around 2006. If you could jump to the next slide. Thanks, Peyman. So management for water chestnut is kind of, again, sort of standard. You can do herbicide or you can get out on the water and pull it manually. But either way, you need to do that sort of monitoring and surveillance to find populations and track the efficacy of your management efforts. And so Ducks has been involved in management in a number of places in Ontario. But as the technology has become accessible, we've begun to use drones to do that surveillance and monitoring. Like right now, we've been able to fly drones over an area where there is chestnut, like the picture on the right, and kind of delineate areas for our on the ground or on the water management team to go and pull chestnut, but they still have to work their way around and find those individual plants, which brings us to this tool that is being developed with Saiwa. And I will pass it off to you, Peyman, to get into the awesome details of this tool.
Thank you very much, Matt. Okay, as Matt said, Ducks Unlimited Canada specialists go out with their drones and collect drone images over vast areas to identify invasive species. The collected images then need to be inspected, and the scattered and the very small and tiny European water chestnuts should be detected in these parts. Even though the field work is very fun and interesting, this part of the job can be very tedious. Every trip, around 1,500 images per day will be collected, similar to this image that you can see here. So if you consider only 2 minutes per image for analysis of the data, then it takes 50 hours for analysis of the data of 1 day. So the question is there any way to automate this tedious part of the work or not? That's the part that Saiwa comes in help with automation of detection. At Saiwa, we develop all essential tools for fast and easy integration of data-driven solutions for small and medium-sized companies, or for institutes such as Ducks Unlimited Canada that don't want to deviate from their main mission. Basically, we have identified two major challenges for companies that want to be beneficiary of advantages of AI services. If they outsource the customized solution, the solution can be effective, but it can be very expensive. And if they want to develop it in-house, then they need to have develop all these capabilities in terms of the expertise that they need to develop. Saiwa, since it is the owner of several problem-specific solutions, allows our team to provide affordable and effective AI-based solutions. In a couple of slides, I would like to walk you through the steps of the project, such as the one that we are talking about today. Generally, we go through 6 steps. Step 1 is data collection, that basically Ducks Unlimited Canada shared their drone images of July 19th to July 22nd of 2022 with us, collectively around 4,500 images. That was for the initiation of the project, and after that we need to work on these images. The first part is basically some pre-processing on the data. The size of the data is usually, the images is very large, so we should split them to the manageable sizes that computationally handleable by our code. In the third step, an expert is needed to label the image. In this process, on Saiwa platform, we determine what region of an image includes the species of interest. This is the regions that code will look into that and will learn from the features that are in that specific part. So you can see here that the process of making a polygon around each of these European water chestnuts that are appearing in the image and labeling them initiated with our colleagues at Ducks Unlimited Canada, and then we took over and continued. In the next step, we need to find the appropriate model of our neural network. A model or algorithm can determine the patterns that are embedded in an image. There are so many different architectures of deep learning models, and one of the advantages of our team is that we have a rich pool of these models, and we also the corresponding patterns that each of them can identify. Therefore, we can distinguish which model is appropriate for the project. After choosing the appropriate architecture of the model, we train the model, meaning that we pass the labeled images to the network to learn from them. We provide our models with the data that was generated on July 19th. And in this way, the model learns from the labeled data and what are the patterns that we are interested in. After learning, we need to test our model for unseen images. And the unseen images in our case was the data that was collected from 20th to 22nd. And the model was able to detect 95% of the European water chestnuts correctly and 5% false positives and false negatives. Here is a snapshot of the first version of European water chestnut software capable of detecting the European water chestnuts with the rate of 5 images per second with accuracy of 95%. However, in practice, we learned that there are some obstacles in the way of having a high precision of this detection. Here you can see 3 drone images from water chestnut from 3 different drone heights, the same drone, but different elevation of the drone. You can see that the way that water chestnut appears in each of these is even difficult for an expert to identify them. One solution is to develop protocols of data collections that our colleagues at Ducks Unlimited Canada developed those. But we admit that it's not always possible to follow the exact protocols. The other challenge there is alteration of environmental features such as water, tree, plants, a reflection of sun. You can see reflection of clouds here, and then saturation of the image because of the sun, presence of the sun. And all these things are the type of obstacles that reduces the chance of generalizing the solution. So basically, overall change of landscape, climate, altitude of drone flight increases the probability of mistake, which to address these issues, our solution was twofold. One was that we invited different groups to share their drone images with us to expose our model to more scenarios to increase the generalizability of the model. Also, we implemented a feature known as incremental learning in the software that makes the learning procedure continuous. In this process, we have initially here the drone data that goes to a model, and then detection happens. Sometimes it is correct, sometimes it's wrong. In the process of incremental learning, we embed some modules in the code that can learn from wrong detections, false positive and false negatives, and therefore gradually the model becomes smarter. There are other features that we added to the second version of the code that I will walk you through those as well. So here is a short video of that shows the workflow of the detection of a European water chestnut on our platform. First of all, what we considered was that we made this platform expandable, meaning that other invasive species can add to the library here. Also, this platform can work both online and offline. one person can initiate a new session simply. The user is allowed to change the threshold of confidence. Upload of image can happen by clicking and guiding towards the folder. Then it is possible to observe the drone image on Google Map for basically seeing where we are in each image. Also, this works with both orthomosaic and individual images. The process of detection can happen by clicking a button. Images will upload and it is processed. So after detection, it is possible to view and observe the detected species. One by one, an expert can go through them and verify the accuracy of detection. In case that there is any false positive, such as the one that you see here, by simply the expert can let the code know. And also it is possible to scan the code. Therefore, if there is any false negative, one can identify those by putting a polygon around that. Now, it is possible that if there are some mistakes, retrain the model. The rate of accuracy is around 95%, but that 5% can be relearned by the model, therefore gradually becomes smarter. The output is in the format that is compatible with ArcGIS. Therefore, the coordinates of all detected species can be listed in a way that can input to ArcGIS as well. Multiple users can use this platform at the same time. One admin, but so many users. And also admin has access to the computational cost of the calculations. So overall, we have a platform that is extendable for multiple species, works both online and offline, easy upload of images, observe drone images on Google Map view, easy initiation of detection process, visualization of the results, review of the results, easy marking of false positives and false negatives can happen on this platform, easy activation of retraining process, global coordinate generation compatible with ArcGIS, multiple user platform, and also track of computational cost. Next step, this summer we will be field testing the tool with our management on Wolf Island. We will fly the target areas, process the imagery through the tool, and put the output coordinates onto the contractor's GPS. This way we save on survey time and we can gauge the success rate of the model. So far, this has been what has been accomplished, but potentially we can expand it to other invasive species of interest. We can also do the identification of dominant vegetation communities and mapping of those. It is also possible to extend and expand the detection to wild animals and objects if it is interest of anyone. At the end, on behalf of Saiwa team, I would like to thank Ducks Unlimited Canada for their friendly approach and share of knowledge, and also Green Shovel's collaboration for financial support of this project. And thank you for your attention. If there is any question, I would be happy to answer.
Thank you so much. What a great presentation. It's really great. This session is, a lot to do with a lot of the projects going on for detection. And this is certainly very innovative thinking, which is really great to see. So Thanks so much for that. And of course, too, we have our own European water chestnut monitoring program in the Welland River. So that's yeah, it's a big project for us too, and it's difficult to get it out. So yeah. Yeah, so we'll open up the floor to any questions. We have a couple of minutes. I did have one actually on if you've had any, issues with jurisdictional restrictions or any sort of permitting issues with using drone technology, maybe potentially in like private property, anything like that.
Matt, I'm looking at you to answer.
Yeah, so as far as I know, we haven't yet. I'm not the one that does the actual drone flights, but I think it's so far we haven't had any issues getting permission from any landowners to do any drone flights.
Well, that's good to hear. Yeah, no, I suppose it's all for good. So that's glad to hear that. Yeah, so we can give it another minute or two for questions. I know it's late in the day, so I think everybody's feeling a little tired, which is fair enough. But yeah, no, that was a great presentation. So I'll leave the chat open if you don't mind keeping an eye on it. We'll just keep on moving along, I guess. Thank you so much, both of you, for the presentation. It was really, really great to see that work.
Thank you for the opportunity. Thank you.
Cheers. Yeah. Alrighty, our last presentation for today is from Adam Perez from the U.S. Geological Survey. And so, yeah, I think we have an interesting chat coming up. So looking forward to it. Thanks, Adam.
Thank you so much for inviting me today. Let me share my screen real quick. And voila. I think everything should be in order. Can everybody see my slides?
Yeah, looks good.
Excellent. So again, I really appreciate, I know it's late in the day, so a lot of pressure on me today, but again, really pleased to be here today to talk about our little bit innovative, a little bit risky line of work, specifically dealing with a lot of pesky pests around the Greater Everglades of Florida. So I wanted to start off by saying that I'm a biologist at the Conservation Genetics Laboratory led by my PI, Maggie Hunter. It has been an honor to be given the task to explore the feasibility of genetic biocontrol applications for, assisting with the management of the invasive Burmese Python population in the Greater Everglades of Florida. For those of you that do not know or not aware of this issue, the invasive Burmese python has been established in southern Florida for more than 30 years and occupies tens of thousands of square kilometers of wide-ranging habitats in this region. What is quite a tragedy is that these invasive large constrictors have been implicated in the severe loss of mammal populations in this region, with up to 99% reduction in many native populations. To date, their large population size and high predation rates could potentially speed the extinction rates of imperiled or at-risk native species. So a lot of efforts are ongoing to mitigate these impacts, but the need to complement these biocontrol methods is quite urgent in the Greater Everglades. And this is a region of not just domestic, but also international significance. It's actually designated a Ramsar site, and restoration efforts from the fauna to the hydrology of the regions are of high domestic priority. So now, why do I say that it is time to look at other approaches to dealing with this problem? The reason is that despite being large snakes, pythons are very difficult to spot, and the region itself is very inaccessible. So their coloration behavior basically leads to traditional detection methods to being as inefficient as 1% when you rely on human visibility alone. And again, like I said before, the inaccessibility of the environment makes it also very complicated to implement removal and other traditional biocontrol methods to extirpate these pesky invaders. So this is where we want to give a voice to innovative approaches, and one of which I'm very interested in as a synthetic biologist is genetic biocontrol, which I define as the manipulation of genetic material with the intent of decreasing the ability of an invasive species to thrive in the non-native environment. What is very interesting about this approach is that it does not rely on direct human interference. So it is designed to be also species specific and to provide something known as landscape biocontrol that it can actually regulate on its own. So one of the most interesting things to look at is that many genetic biocontrol approaches focus on distorting sex ratios. For example, you can see in this slide how when you look at inheritance, specifically in gender ratios for a lot of animals, it is inferred to be 50-50. So you can have 50% females, 50% males. Some genetic biocontrol technologies actually can focus on distorting that so that you can promote a single sex population over generations. So on the right side, many of these strategies, particularly in insects and small mammals, try to push for the disruption of, specific sex chromosomes during sperm or egg development, so that progeny of a single sex is the only one that is viable. In this case, only the male population, and this is done with the intent that in the future, population collapse could happen because of the inequality in male to female populations. So again, that's just a general example of what we're hoping to emulate for invasive reptiles. But again, easier said than done. And it is even more true when it comes to genetic biocontrol, because we do not only focus on the technical aspects, which by themselves are already pretty laborious. So genetic biocontrol cannot happen in a vacuum. And what do I mean by that? Well, we actually need to develop a framework that needs to be understood to have many different components that work together. And, you also have to be cognizant that results are not going to happen in the span of a year. So this is something that is planned over many years, and it includes many hard stopping points along the way of each phase for discussion and co-production by a lot of the shareholders, namely scientists, the general population, and social scientists to discuss. So in bullet point 1, which is what I'm going to be focusing on today, it's what I like to shorten as the technical part, where you actually try to see whether or not it's feasible to genetically explore these methods in an organism that is not known as a model. It's not a fruit fly. It is not a white laboratory mice. It's something that is actually very difficult to work with, because very few people have had the opportunity to research this in the past and to manipulate it genetically. So there's a lot of research that goes along encapsulating the selection of target genes, genetic testing, validating how these changes affect behavior, and so on and so forth. And number 2, which is also an interest of our laboratory, is that a lot of people are not comfortable with anything that has to do with genetic manipulation modifications. So you need to follow a model of open science, where you have shareholder engagement in our case with federal, state, tribal entities, but also the general population at large, because you want to ensure them that you're ethically looking at the problem, you're actually looking at safeguards against any consequences, and obviously unintended consequences are very difficult to foresee, but this is something that cannot happen in the back end, and I cannot stress that enough. So point one and point two are the main focus of our lab, but again, many other teams have spent numerous efforts and time exploring other things that are, in my opinion, equally as important. So in this case, I want to stress out and commend other researchers for focusing on things such as population modeling and vital rates, because as I mentioned at the beginning, these snakes are very cryptic, very secretive, very hard to understand what they're doing in the Everglades besides the impact on, those native species. But we need more information on where do they live, how do they behave to weather changes, how often do they reproduce, because you cannot extrapolate 100% what they do in their native range to what they're doing in this new environment. So this is critical whenever we talk about genetic biocontrol, because if you have the know-how in terms of the genetics, if you do not understand how these organisms behave in the environment, it's all for naught. You really need to marry the idea of the whole organisms with the genetics that we're pursuing. On top of that, needless to say, point number 4, risk assessment is critical. This is not just to, calm the general population and that what we're doing is being done with care. But again, there are a lot of things that need to be considered when applying these technologies. A lot of people, for example, have concerns. What would happen if such technology made its way to the native habitat? Do you have safeguards to eliminate any potential negative effects there? Again, there's a lot of things that need to be considered. There's unintended consequences that could happen by manipulating the genes of an organisms, and you need to take all of this into serious account and have multiple safeguards to make sure that things do not go out of control. And last but not least, there's a lot of regulation and permitting that needs to go on. Like in previous talk, you will have to deal with a lot of different people that have different outlooks, different value systems, and you cannot just fly a drone in anybody's property. Likewise, you cannot just implement a program of this magnitude in anybody's backyard. So you do need to take into account the regulation and permitting as well. So what I'm trying to say here is that projects of this nature need to incorporate more than just the science, technical know-how to actually have a chance at succeeding and supporting all these biocontrol methodologies that are in place today. Like I said previously, we're going to be focusing mainly on the technical side of things, a little bit of updates of what we have done and where we hope to go next. So again, very difficult to work with large snakes. As you saw in the previous pictures, they're not tiny. And there's a lot that we do not know in terms of how to manipulate them genetically. Fortunate for us, there's a lot of pathways to pursue that do not entail raising snakes and having to deal with a lot of questions of why we're doing that. We are very interested in using models based on culturing some cells of these Burmese pythons. And these cells are what we call primary cells. And we just culture them directly from tissues of recently deceased snakes, grow them in a petri dish, and these cells have not been modified in any way whatsoever. So they're the best representation of how these genetics changes could translate to a live organism without utilizing the live organism. And it's actually quite nice to see that you can grow them, expand them, and do experiments ad infinitum almost, and they look quite nice. These are actually heart muscle cells or myocardiocytes that we grow in our laboratory, which is a biosafety level 2 tissue culture laboratory in the Wetland and Aquatic Research Center that I'm part of in Gainesville, Florida. And these cells are first characterized, so we want to know how they behave, how they grow. So that is very important to proceed before we do anything more in depth. So after accomplishing this fundamental work, then we're ready to go to the next question. And that is, nobody has genetically modified these snakes like they have done with fruit flies or bacteria, white mice. How do we start this? Well, first and foremost, we need to know, can we introduce any foreign genetic material into these cells, and how? There's a lot of methods to accomplish this. Some of those are very interesting, such as electroporation, utilizing electricity, but you could see how this could be a little bit more challenging to do once we get to the point a couple of years down the road to work with live snakes. So we decided, can we do that utilizing something simpler, like using lipids or oils that would encapsulate this foreign genetic material? And you have to remember that cells are basically oil-based, and you could use that to your advantage to introduce things that otherwise are not part of the cell that you're targeting. And the most logical thing is, let's introduce a gene that can produce something that we can track or see. And everybody likes fluorescence, so we decided, can we use lipids, aka oils, to introduce a gene that produces something that fluoresces, a protein that fluoresces, aptly named green fluorescent protein? And the answer is yes. So we were quite excited to accomplish this, because not only were we able to introduce this genetic material, but this genetic material was able to be functional inside of the Burmese Python cells. And again, everything is about optimizing the procedure, because again, nothing is as simple. You need to balance out whether or not it's toxic to introduce these foreign genetic elements. How toxic are these foreign lipids? So there's a lot of balancing here to see how long you need to, expose these cells to this concoction to result in the maximum transformation, we call, of the cells. Very happy to see that, but then is that all we need? Well, not quite. I wish it was, because it's actually pretty good cool to see these cells glowing in the dark. So what I mean by that? Well, originally I said there's very little work done in Burmese pythons. So what makes us confident that we can actually work on them now when a lot of people didn't even bother to do that? Well, let me tell you about CRISPR-Cas. CRISPR-Cas revolutionized everything in terms of how we can gene edit practically any organism that we have a genetic sequence of, even if you know very little about said organism. So CRISPR-Cas are basically these molecular scissors that you can program to target virtually any sequence in an organism's genetic code. So naturally, we thought if we want to introduce genetic biocontrol elements, then we need to be able to genetically modify the organism. And what better than CRISPR? So we were able to introduce a gene that produces a protein that fluoresces green. The next logical step is, can we introduce a CRISPR construct that is a lot more complex than a gene? It has proteins, it has nucleic acid like RNA, DNA sequences in it. Can we do that the same way that we did that with the green fluorescent gene protein? We'll see. So before we did that, we needed to realize, what are we going to target first? There's a lot of options in terms of genetic biocontrol targets, but we need a control. So in 2019, at the University of Georgia, they demonstrated that CRISPR can be introduced into reptilian models as in the same manner that we're attempting to, and you can actually produce characteristics that are traceable. In this case, there's a gene called tyrosinase that is very important in producing the pigments in your skin. It happens also in reptiles, and if you disrupt this gene, using CRISPR or other methods, you can induce a very visible characteristic like albinism. So you can see an albino lizard and one that is your typical brown. These are brown anoles, so very apt here. So this tyrosinase or TYR gene has 3 very specific amino acid residues that are important to carry out its function. We look at the sequence in these brown anoles and the ones in Burmese pythons, and we were able to identify a suitable target in the gene that would disrupt the function of this protein once it's expressed in Burmese pythons. So you don't need to know much about this, except that there's a lot of really nice letters for what we call the DNA nucleic acids. But these 3 H's that I highlight are the important sequences that code those amino acids that are critical for the function of this protein. So in blue, you'll notice that we have a lot of options in terms of what type of CRISPR enzymes we can utilize. The most popular being what we call CRISPR-Cas9, where most of the novelties came from, but we have a suite of CRISPR complexes now to work with, which is quite fantastic. So now that we understood where we wanted to start off, we decided to make a hybrid CRISPR molecule that had a fluorescent marker, because we wanted to know whether or not we can introduce this programmed CRISPR that will target the albinism gene in Burmese pythons. We want to know, does it go inside? And this fluorescent marker called ATO550 can actually be tracked quite easily utilizing some microscopy features and other techniques. And we realized that based on the parameters that we used in our first experiment to introduce that gene that made the green fluorescent protein, we had high efficiency, meaning that a lot of the cells that we exposed to the CRISPR complex, which again, remember, it has proteins and nucleic acids and the fluorescent marker, we had more than 85% of the exposed cells showing an indication that the CRISPR was incorporated successfully inside. Is that the end of the story? Fortunately, no, I wish it was, but I'm very happy that slowly but surely we're progressing to getting these fundamental knowledge out so that we can build upon this technology. Once we did this and we incubated the cells for a little bit, the next step is, did we actually gene edit that albinism/tyrosinase gene? So to do that, we just took the cells out, extracted the DNA, and compared the DNA of the cells that were exposed to the CRISPR complex to cells that were not subject to this gene editing mechanism. And unfortunately, at this stage, we were unable, despite having the CRISPR molecules inside the cells, to induce a change. And we would expect that the sequences that you see here on top and bottom would be different, but unfortunately, they are the same. So now this is where we stand when it comes to the technical side of things, and we need to continue understanding what we can do to improve this process. So the cells that we work with, we need to troubleshoot. They are already pretty old. As much as I wish that they do last forever, we want to make sure that we do not modify them, make them immortal, cancerous. We want them to be as closely related to the organism as possible. So that means that they will eventually become old and not work. And these cells we have been working with for quite a little bit. So the goal is to actually procure younger cells. We call them affectionately Boopy for Burmese Python, which I actually quite like, to test this methodology once more, because the younger the cells, the more amenable to CRISPR edits they are. And of course, we need to test additional delivery methodologies. The lipid oil-based methodology is known as liposuction. So we want to see if we can do that in other ways with different delivery options, different exposure times, different CRISPR concentrations to see if that is the roadblock to gene edit something. And of course, you can always program the CRISPR to target different sections of your target gene, which is fantastic and could potentially be the answer to what we're looking for. Once we successfully gene edit this target that I presented to you today, the tyrosinase, the next logical step is, instead of disrupting, is introducing complete new genetic elements into the genetic or composition of the Burmese Python cells. So we can introduce markers that are fluorescent to track whether or not this works. And we're actually quite excited because once we complete this step, then this opens the door to introduce practically any methodology, including genetic biocontrol methodologies, to work in these organisms. So again, I just wanted to let you know that genetic biocontrol take home is not a simple experiment. It's not something that can be carried out by a single team. You need to have a very extensive collaboration of a lot of personnel with different expertise. And you need to be responsible because a lot of our shareholders are worried with reason that these type of experiments could be risky, could be taken in a negative light. And again, we do not want to repeat a lot of the sad examples from the past where scientists will just present their work to make sure that people buy into what they're doing without really having any input or without hearing their concerns, addressing their concerns, and co-producing these research projects. So again, this is something that goes beyond the life science. You need to, again, incorporate the knowledge by managers, social scientists, tribal leaders, tribal citizens, federal, state agencies, to make sure that this work is done correctly. And you need to be accepting that, at times it is best to not proceed, not just because the technical side might not be successful, but because there's just enough roadblocks, in trying to make sure that people are comfortable with this technology. So you have to be ready to provide these hard stops that, might make a lot of other scientists a little bit nervous to say, yeah, we're willing to stop this if, you're not comfortable with it. So engagement is absolutely critical. I want you to take this home. And again, a lot of things that you need to take into account, like make sure that you protect and prevent unintended consequences as much as possible. You do not affect the native populations that might be related to your target organism, risk assessments, and making sure that you're responsible above all. So with that said, again, I want to thank everybody for listening in today and to the organizers for inviting me to give this presentation. I am more than happy to answer any questions or have a discussion about any of the parts that I presented today. And again, I guess to those of you that have it in your country, happy Valentine's Day, I guess.
Thanks so much, Adam. No, that was a great way to end the day, and I appreciate your ending there on engagement and everything. So that was a really, really nice way to end things. We do have one question, and I will be mindful that we are running out of time. So somebody has said that they have read that it may be possible that when using the CRISPR function to sometimes have slight errors in the ends of the gene sequence that is being introduced. Is that a concern to your research at all?
Absolutely. And that is something that we need to be aware of because, again, our bodies and those of the animals and plants that might be targeted are very complex. And, you could have the case where CRISPR itself will target your gene of interest, but again, there could be similarities in other genes or sequences inside of your organism of interest that could be affected. So again, there's a lot of work to ensure that CRISPR only targets what you intend to, but you cannot guarantee that 100%. Our bodies are too complex, and unless you sequence everything after your gene editing approach, it is very hard to say that that's all you affected. So yes, this is something that needs to be explored, not just for this project, but for any CRISPR-based project. So that's a really good question, because it is indeed a concern.
Thanks so much. No, really appreciate that. And yeah, with that, I'll pass it back to Maddie. Thanks so much, everybody, for joining this afternoon.
Thank you.
Awesome. Well, that's a wrap on day 3 of our Invasive Species Forum. Thanks, Jenna, for moderating this session, and thank you to all of our speakers for their excellent presentations. We had an absolutely jam-packed day of invasive species learning, from oak wilt and forest health all the way to monitoring and policy updates, and we hope you enjoyed it all. If you missed any of the sessions, we'll have all the recordings posted over at our YouTube channel with speaker permissions. A lot of them are already up and available right now. We have some exciting things planned for our final day tomorrow, so make sure you stick around another day and find out what we got going on. We will be kicking things off just before 9 a.m., so we'll be meeting at 8:55 a.m. Eastern Standard Time tomorrow for the Invasive Species Centre's award ceremony to recognize some amazing individuals that are working to help keep the land and water in Canada free from invasive species. So really looking forward to celebrating this year's nominees and winners. We also have a really fun game of invasive species trivia Kahoot. So join us tomorrow at 11:45 a.m. with your competitive hats on to show off your invasive species knowledge and potentially win a prize. So that's it from me, everybody. I hope to see you back tomorrow at 8:55 a.m. Thank you so much.